Hi everybody, we're back again and we're here to talk about learning goal number 11, and we're going to again keep on moving through our genetic traits unit. This is our last learning goal in that unit, and we've been moving beyond Mendelian genetics, and today we're going to focus on sex-linked, sex-limited, and sex-influenced inheritance and the way that changes our predicted offspring ratios. Now, in order to understand sex-linked traits, you have to understand the X and the Y chromosome. See, the thing is the Y chromosome is much smaller than the X chromosome. And as I'm assuming you know by now, ladies, you have two X chromosomes. And gentlemen, you have one X and one Y. So that really changes things for you guys, because ladies, you have two complete X chromosomes which both contain all kinds of genes. Gentlemen, this Y chromosome, it's kind of like a little stub. It really doesn't have very many important genes. It has one very important gene to you anyway, which is your SRY gene. And that initiates sex determination in humans. It's what makes males males. So now that we understand the way the chromosomes work, we can see how that influences offspring and on certain traits when those traits are contained on the X chromosome. So one of the most important things to understand is that these genes that are only present on the X chromosome are going to show a different pattern inheritance than normal autosomal inheritance. We use a certain kind of notation when we're talking about this. The X chromosome will show in superscript either a little a or a big A. The little a shows the recessive one, and the big A shows the dominant allele. This is a way of showing that this is a trait on the that's on the X chromosome. Therefore, when you write, males will only have one X with whatever letter, and then they'll have their Y, which won't have any letter on it, because that Y chromosome will not have that particular gene or that allele. Okay. Now, the other thing, let's talk about how we figured this out. There was this guy named Thomas Morgan, back in 1910, was working at Columbia, and he had this fly lab, and he was crossing all kinds of fruit flies. So you'll see those are his fruit flies here in all of his jars. And he was really excited because he found this new white-eyed mutant in the Drosophila. And when they looked, he started looking at the differences in phenotypic ratios of reciprocal crosses. So let's pause for a minute and answer the question in italics. What is a reciprocal cross, and why would we expect phenotype ratios between reciprocal crosses on autosomal traits to be the same? Go ahead and give that a shot and pause me. Okay, hopefully at this time you've already written a little bit about reciprocal crosses, and hopefully you've written down that reciprocal crosses involve a cross where maybe the male has the trait of interest and the female doesn't, and then you do another cross where the female has the trait of interest and the male doesn't. So for our example we've been talking about today, we start with a red-eyed female crossed with a white-eyed male, and then a white-eyed female crossed with a red-eyed male. Now normally we expect to get the exact same kind of offspring when we talk about autosomal crosses, and that's because sex does not affect the results of autosomal crosses. But that's what got exciting with Thomas Morgan. He noticed some different kinds of results with these white-eyed fruit flies. So when we're working on these problems, it would help if you knew the signs for males and females. Females kind of look like a stick figure, and males look like a circle with an arrow going up at an angle. And you'll see those symbols show up a lot throughout these problems. Now, if you look, Morgan discovered that when he did his reciprocal cross, these were his results. Now I want you to go ahead and turn to the person next to you, and you've got this same diagram in your notes. Talk about what you see, and then you could pause me. Okay, now that you've talked about that, hopefully you've seen that the reciprocal crosses between red and white eyes for fruit flies were not the same. So when they had a red-eyed female and a white-eyed male, you got all red-eyed offspring. But But when you had a white-eyed female and a red-eyed male, you got some red and some white. And so Thomas Morgan was curious about why that happened. And he came up with a hypothesis. So his hypothesis was that males only have one allele, and it's on their X chromosome. And he also thought that maybe that allele is totally, that locus or that position for that gene is missing on the Y chromosome, and that's why you see these differences in reciprocal crosses. Let's look at this diagram to help you map it out. So again, if you've got a red-eyed female, she might have 2 copies of the red allele. Cross with a white-eyed male, he's got 1 copy of the white allele, and then there's this male, Y chromosome that doesn't have anything. Don't worry about it. So the only thing that the female can give is the red allele. The male can either give the white allele or the Y chromosome. So that means that, again, everybody's getting the red from the mama, and the 50% of the offspring will get the white allele, and 50% of the offspring will get on the X chromosome, and 50% of the offspring will get the Y chromosome. So these red females are all carriers, and these red males don't have the allele at all because they just have the 1 copy, and it's the red allele. This white allele never made it to them because they got the Y chromosome instead. Okay, so that ends up with all red offspring. Let's look at the reciprocal cross. In order to be a white female, you have to have 2 copies of that white allele. The male has 1 copy of the red allele. So again, everybody's going to get the white allele from mom. And from the male, you're either going to get the red allele or you're going to get the Y chromosome. Now what happens is that means half of the offspring will be red. They are carriers for that white allele, but they still look red. And half of the offspring will be males, and they will all be white. Again, different possible phenotypic ratios depending on whether which parent was female, or whether the female was white or whether the male was white. Based on this information, I want you to go ahead and try to answer those 3 questions in italics on your Big Ideas packet. Pause me and come back when you're ready to go over the answers. Okay, so hopefully you've answered the 3 questions. First of all, I wanted you to explain why the male fruit fly, but not the female, demonstrated the white-eyed trait after the cross between the F1 generation. So when we're looking at this, we're saying to ourselves, females with 1 white allele will not show the trait. But males with 1 white allele will show the trait. So it requires more for the female to actually show the trait. It requires 2 alleles. Males only need 1. Let's think about this. Is it even possible to have a white-eyed female? If so, what cross would you perform? You can get a white-eyed female as long as the mother is a carrier and as long as the father has white eyes. We've seen examples of this. Again, here's our carrier red-eyed female crossed with a white-eyed male. This white allele can come from the mom, and this white allele can come from the dad, and that's how you end up getting a white-eyed female. Now these mutations are pretty rare, and Morgan's white-eyed male fly probably arose because of mutation. So why was it more likely that we got this mutation in a male than in a female? Well, I'm hoping that you thought about the fact that a mutation would only have to show up 1 time, 1 allele on one of the 2 chromosomes in a male. But for it to happen in a female, you'd have to have 2 identical mutations show up at the exact same time on both chromosomes in order to have that white-eyed phenotype show up all at once. All right, let's continue talking about sex-linked traits. One of the things that you need to know is males only have 1 X chromosome. Because of that, they can't be homozygous or heterozygous on an X-linked gene. We call this hemizygous. because there's never going to be any alternative allele present. There's only going to be one allele at a time. The concept of dominance and recessiveness is pretty irrelevant when it comes to males on sex-linked traits. Now, lethal X-linked recessive disorders we are only going to see in males, because females are the only ones that are going to be carriers, and you're never going to see a female who is a female pass it on. You aren't going to see females and males mating together with this trait. For example, if it's lethal, a mother who's a carrier might give it to her son. But in order for a female to end up having it, the father would have to have the trait as well, which would mean, and that's not going to happen because the dad is going to die before it can father any children. Take a second and answer the next two questions in italics for me, please. And pause me when you're finished. Okay. Before we talk about those two questions, let's look at some examples of X-linked traits. Color blindness, which again is not lethal, so there may be somebody in this room that has red-green color blindness. Hemophilia A and B, also muscular dystrophy. These are all common X-linked genes. Again, we're seeing them more often in males than we do females because it is easier for a male to get a trait like this because it only requires one of these recessive alleles to show up in a male, where females would require two. Let's check to see if any of us are red-green colorblind. Look at the screen. What do you see? If you see a number 74 right here, then you are not red-green colorblind. If you cannot tell the difference between this number 74 here and the rest of this circle, you may be red-green colorblind. This is super common because, again, this is not something that is lethal, and it's not something that really drastically affects us. So we will see sometimes even females with red-green colorblindness, though it is more common in males. Now let's look at the answers to your questions. What is the probability that a male will inherit a sex-linked recessive disorder if his mother has a disorder and his father is normal? What I would expect to see, and you could have picked different letters than A, but that's what I picked, a mother who has the disorder, so she's an X little a, and a father without the disorder that's normal is an X big A, Y. So it really comes down to the father. The mother is going to give an X little a no matter what. But the father's either going to give an X big A or he's going to give a Y. So obviously the male is the only one who can have the chance with this out of this set of parents and have the trait. So it's a 50% chance that this offspring will have the trait. Alright, what's the probability that a woman who's a carrier for a recessive genetic disorder will have a son with the disease if she marries a non-affected male? So we have our mother, who's a carrier this time, and a father who does not have the trait. Now, if we assume they're having a boy, which it kind of sounds like it based on the question, the answer is going to be 50%, because if we assume they're having a boy, we know the father's giving a Y. Then it just comes down to the mother. If she gives a big A, they won't have the trait. If she gives the little a, they will have a trait. So it's a 50% chance that their son will have the trait. Really, it's a 1 in 4 chance that any of their kids would have the trait. Okay, let's talk about two other examples where sex influences genotype and phenotype ratios, just phenotype ratios. One of them is called sex-limited inheritance. That's when the expression of a certain phenotype is absolutely limited to one sex. If you aren't of a certain sex, you cannot have that trait. Now, sex-influenced inheritance is different. It is when the sex of an individual influences the phenotype, but both of the sexes can show it, they just might show it differently. So sex-limited, the best example we have for that is milk production genes in mammals. It doesn't matter what genes you have, If you are a male, you are not going to produce the genes to make milk. You are not going to in your mammary glands. Now, but females will. With sex influence inheritance, another good example we have for that is baldness. Now, baldness acts like it's a dominant in males, but recessive in females. So that's a little bit weird. So if you look at this chart here shortly, we see that in a female, she has to have 2 copies of the allele in order to become bald. With a male, they only need 1 copy of the allele to show baldness. So again, the genotypes are not affected because these are on autosomal genes, but it is the phenotype that is affected. The reason is because these expression of these autosomal genes depends on the hormone constitution of the individual. So the hormones of the surrounding organism, whether they're male or female, are going to influence the way the gene is expressed and therefore the phenotype. Go ahead and try to answer the last question in italics for me and pause me when you're ready to go over the answer. Okay, so will the phenotype ratios be different for a sex-limited or a sex-limited trait than for a normal Mendelian trait? What about the genotype ratios? Well, the answer to this is the genotype ratio should be exactly the same. It's autosomal. The genotype is not affected. The phenotype ratio, however, will be different because the number of individuals who actually express or show that trait is going to depend on sex. I hope this helped.